Everyone, and thanks for coming. It's my pleasure, it's my true pleasure to introduce Emma for her exit seminar today. Emma hails from Meaford, Ontario on Georgian Bay and still spends her holidays helping her parents in the bakery. In fact, if you're looking for a good source of cinnamon buns, then Emma can recommend a great place for you. Emma did her BSc in biology at Queen's and worked in my lab as a SWEP student in the summer of 2017. When she became the ace of DNA extractions. She loved DNA extraction so much that she chose to do a 5-7 thesis with me. That project involved an examination of geographic variation in genes related to immunity in an Arctic seabird, the black guillemot, and added PCRs and DNA sequence analysis to her repertoire. She did such a good job with that project that I invited her to do a master's thesis on population genomics of another Arctic seabird, the Glacous Gull, which she'll be sharing with you in just a minute. Emma has been a true joy to have in the lab, always cheerful and always game to try new things, whether it's fieldwork on a remote island or a new method of DNA analysis. During her time in Kingston, Emma also became the queen of finding new apartments, packing, moving, and unpacking. I think she had to move apartments at least 6 times during her time with me. She's going to be sticking around Kingston after she defends her thesis, working for me doing more DNA extractions for a few months. And I think that requires her to move apartments yet again. So without further ado, Emma's going to tell us all about her project.
All right, thank you. Hi, my name is Lachance Linklater, and I'm going to talk to you about my master's project today, population genomic structure and hybridization of Glacous Gull in the Canadian Arctic. So the Arctic is currently experiencing unprecedented climate changes. It's expected to experience greater increases in sea surface temperature and average precipitation than the global mean. And the 2014 IPCC report estimates that Arctic sea ice has reduced by 3 to 4% per decade since 1979. And some current estimates suggest the Arctic might be ice-free for parts of the summer as early as 2037. Anthropogenic climate change is having and will continue to have severe impacts on the circumpolar Arctic. Coupled with the changing climate are other threats. The reduction in sea ice represents an unprecedented, or represents an opportunity for unprecedented Arctic access. And increased shipping is just one example of the ways in which the Arctic might be exploited in the future. For instance, in Nunavut in the last decade, Nunavut has experienced a 400% increase in pleasure craft tourism. And for shipping companies in the Northern Hemisphere, it's expected that trade routes through the Northwest Passage will be shorter and cheaper than current routes through the Panama Canal. This increase in shipping can have all kinds of detrimental effects. Increased shipping could introduce invasive species through ballast water, increase mortality of mammals and birds through ship strikes and accidental entanglement, could disorient migratory species through marine noise and lights, and most significantly, increase the severity and the frequency of oil spills. Climate change can also shift natural species ranges, bringing new species into Arctic territories. These shifts in natural species ranges could increase predation on Arctic species, such as what you see in the picture here, could introduce pathogens that Arctic species don't have any natural immunities to, can increase competition for already limited habitat and resources in the Arctic, and might promote hybridization between previously isolated species. The protection of ecosystems and species are both incredibly important forms of conservation. Ecosystems can be protected through things like the development of federally protected areas. For instance, in 2019, Canada had 12% of terrestrial areas and inland water, and 13% of marine and coastal areas federally protected. Indigenous land management is also a very effective form of conservation. A study by Schuster et al. In 2019 found that Indigenous managed land in Canada, Australia, and Brazil was more vertebrate rich and hosted more threatened vertebrate species than federally protected areas. Species can also be protected through federal and international management plans. These may include things like hunting restrictions, breeding programs, and population monitoring, among other things. The conservation of ecosystems and species tend to get considerable attention, but the conservation of genes is less commonly discussed. Effective conservation has to conserve 3 levels: ecosystem diversity, species diversity, and genetic diversity. Genetic diversity has long been considered an essential component of a species' evolutionary potential, as it's the foundation for long-term population viability and for adaptation. Population size also has an impact on genetic diversity, since small populations tend to have less genetic diversity, experience more genetic drift, and may experience more inbreeding depression. And this can lead to an extinction vortex, which can be exacerbated by environmental changes. The maintenance of genetic diversity, therefore, is critical to a species' ability to respond to climate change. Genetic diversity can be geographically structured. So if we consider, so population genetic differentiation therefore is the process where 2 populations can become genetically distinct. So for instance, if we look at this population of gulls that we have here, we consider this one population. Over time, it might divide and become 2 populations with migrants between. These 2 populations wouldn't be considered genetically differentiated as long as sufficient gene flow was maintained between them. If a barrier to migration disrupts gene flow, then over time, these 2 populations might begin to diverge. And this barrier could be something obvious and physical, probably not a brick wall, but maybe something more along the lines of a mountain range. But these barriers can also be less visible. For instance, just distance enough might be sufficient to create population genetic differentiation if the distance is far enough that the species isn't able to cross between the populations. And given time and lack of genetic diversity, given time and lack of gene flow, these 2 populations might become genetically differentiated as well as geographically differentiated. I'm using these blue and green colors to show population genetic differentiation, but this might not always be very visible. It could be visible with things like morphological or behavioral differences between populations, but it could also be a purely genetic signal that isn't visible to an observer. Population genetic differentiation is promoted through time, isolation or lack of gene flow, drift, and divergent selection. And differentiation tends to decrease through gene flow and balancing selection. Now, if these 2 populations of gulls in this case reconnect, but they haven't developed reproductive isolation, then they could produce an offspring that has genetic similarities to both of the parental populations. And if these 2 species of gulls in this case, or 2 groups of gulls, are considered different species, then this offspring would be considered an interspecific hybrid. And hybrids can exist anywhere on the spectrum from complete sterility, often due to chromosomal asymmetry in the parental species, to greater fertility than either of the parental species. And hybridization is considered fairly frequent in birds. Some estimates suggest that 9% of bird species hybridize with another. And some work by Price and Bouvier in 2002 found that, or estimated, that it might take 10 million years of species divergence before hybrids are infertile, and 17 million years of species divergence before hybrids are considered inviable. There are some benefits to hybridization. For instance, first generation or F1 hybrids may experience something called hybrid vigor, where the F1 has higher fitness than either of the parental species. Hybridization can also generate new species, and this is called hybrid speciation. An example is the golden-crowned manakin, which is a bird in the Amazon. It was recently discovered that the golden-crowned manakin originated as a hybrid between opal-crowned manakin and snow-capped manakin. But hybridization can also provide a source of novel genetic diversity that a declining species maybe doesn't have access to, and this can create a form of genetic rescue. There are also consequences to hybridization. So even in species where the F1 has hybrid vigor, it's fairly common for later generations of hybrids to decrease in fitness, and this is called outbreeding depression. Hybridization can also eliminate evolutionary adaptations to the local environment through genetic admixture, and eventually lead to the extinction of a population or species. And if hybridization is occurring between a threatened species and an invasive or introduced species, differences in stability between the 2 can often lead to the threatened species being demographically or genetically overwhelmed. And in the example photos I've got here, about the mallard duck and the gray duck, mallard duck were introduced to New Zealand in the 1860s, and they began hybridizing with the native gray ducks. And over time, through hybridization and overhunting and habitat reduction, mallard ducks have essentially eliminated any pure representatives of the gray duck in New Zealand. So there's several conservation strategies that involve the division of conservation units in order to appropriately manage distinct genetic populations. And Funk et al. In 2012 suggested a hierarchical method where you first use all of your genetic loci and look for population genetic differentiation, and those differentiated populations are considered evolutionarily significant units, or ESUs. Then Funk et al. Suggests doing an outlier analysis and finding neutral loci and putatively adaptive loci. And then populations that are differentiated with neutral loci are designated as management units, or MUs, and populations differentiated at putatively adaptive loci are considered adaptive units, or AUs. High-throughput sequencing has blurred the lines between model and non-model organisms and has allowed the generation of incredible amounts of high-quality DNA for a relatively low cost. Double digest restriction site-associated DNA sequencing, or DDRAD sequencing, is a method of high-throughput sequencing where DNA is digested by a set of restriction enzymes, ligated by a set of barcoded adapters, digested again with another restriction enzyme, ligated again with more barcoded adapters, and then amplified to produce many, many copies of the same fragments. DDRAD sequencing depends on high-quality samples, but it can produce millions of fragments of DNA for analysis. It's incredibly important to create proactive and informed management strategies to protect vulnerable species. And the goal of my master's project is to explore population genetic differentiation and inform the conservation strategy of Glacous Gull in association with Environment and Climate Change Canada. So what are Glacous Gull? Glacous Gull are a large species of migratory Arctic seabird. They're actually the second largest species of gull in the world, with up to a 6-foot wingspan. They are aggressive generalist feeders. They'll scavenge from carcasses, steal food from birds and other animals, hunt and kill chicks and adults of other seabirds, and like you see in the picture, steal eggs from nest sites. They have a circumpolar breeding distribution, and they tend to nest at low densities, which is pretty typical for an apex predator. They're also monogamous and very committed to their breeding partner. So a study at Coates Island over 22 years found just a 9% divorce rate in Glacous Gull. And they're also philopatric. In this case, they both return to the site where they were born to breed, and they tend to return to sites that they've been successful in the past. They're also a long-lived species, living for 15 to 20 years, and it takes 4 years for them to become reproductively mature, and that's typical for large gulls. Glacous gull are currently listed as least concern by the IUCN, but they have experienced declines in Canada, Iceland, and Svalbard. And it's important to note that there's no conservation units designated for Glacous gull's protection in Canada. These declines in population may be due to a range of different threats, including the bioaccumulation of environmental contaminants. So Glacous gulls, as apex predators, bioaccumulate high levels of environmental contaminants. And Glacous gull in Canada actually have higher contaminant loads than any of the other seabirds in Canada. And high concentrations of these contaminants have been linked to all kinds of negative effects, including reduced reproductive success, reduced adult survival, weakened immune function, altered hormone production, and altered breeding and nesting behavior in Glacous gull. Declines may also be due to other things, such as avian cholera. Glacous gulls have been recorded feeding on eiders, which died in cholera outbreaks, and this might be contributing in some areas. Glacous gull are also harvested for their meat and eggs in North America, Iceland, and Svalbard. And this harvest isn't super highly regulated. For instance, in Alaska, nearly 17,000 non-species specific gull eggs are harvested every year. This makes it really challenging to track the impact specifically on Glacous gull in Alaska. And in Iceland, it's estimated that 15% of the population of Icelandic Glacous gull are killed every single year. And this might seem really severe, but it's also important to note that harvest rates have been decreasing in Iceland since the 1990s, but the population of Glacous gull continues to fall. Glacous gull are also potentially declining due to competition for resources. For instance, in the Svalbard archipelago, specifically on Bear Island, Norway, Glacous gull have been competing with great skua, which you see pictured there. And great skua were introduced to Bear Island in the 1980s, and since then, Glacous Gull have experienced a 65% decrease in breeding pairs. So it seems like competition there with the great skua has a big component in the declines. And finally, hybridization might play an aspect in some places. Specifically in Iceland, Glacous Gull have been known to hybridize with herring gulls, and the herring gulls were introduced to Iceland in, I think, around the 1920s. And so declines may have been partly coming from hybridization since then. So yes, hybridization, let's get into that aspect. So Glacous Gull are a recently diverged species, and they're nested within the herring gull species complex. So this species complex contains 22 to 23 species of white-headed gulls, and some of them have diverged as recently as within the last 30,000 years, since the last glacial maxima. And due to this really recent divergence of this species complex, reproductive barriers between the species are very, very weak. So Glacous Gull are known to hybridize with Glaucous winged gull, American herring gull, European herring gull, and other species as well. And it's important to note here that not all organizations consider the American herring gull and the European herring gull to be separate species. So I am kind of taking a side in a bit of a conflict here, but that's the taxonomy that I'm going to use for the rest of the presentation. And so 2 years ago, I started to get interested in this aspect of hybridization between Glacous Gull and other species. And I started to think about how easy it would be for me to have Glacous Gull hybrids in my samples that I didn't even know about, sort of invisible hybrids. And since all of my samples were blood or tissue, I didn't have any way of going back to look at the morphology of the sampled birds. So I had no way of identifying if they were hybrids there. And I got excited by this idea that I might be able to recognize hybrids using genetic techniques. And so, of course, I joined a passionate Larophile community on Facebook called North American Gulls to become better acquainted with identifying hybrids, seeing pictures of hybrids, and getting to be a part of the discussion around gull hybrids. And what I realized was I was getting involved in a huge mess. So I've created a bit of a chart to help map out the mess for you. I'm just going to adjust this. There we go. Okay, so on the left-hand side, I've got Glacous Gull, of course, the most beautiful of all the gulls. And on the right-hand side, I've got American herring gull, Glaucous winged gull, and European herring gull. So these are the three species that Glacous Gull hybridize most readily with. And in fact, hybrids between these species are so frequent that passionate Larophiles actually have their own names for these hybrid crosses. So for instance, Glacous Gull American herring gull hybrids are called Nelson's gulls, Glacous Gull Glaucous winged gull hybrids are seaward gulls, and Glaucous European herring gull hybrids are called Viking gulls. And furthermore, American herring gulls also breed with Glaucous winged gull. These are called Cook Inlet gulls. And as I mentioned before, American herring gull and European herring gull aren't always considered to be distinct species. So they also hybridize, but whether or not you consider those to be interspecific hybrids is up to you. Now, this is where things start to get a little bit wild. So I'm going to introduce you to some of the other members of the species complex: the kelp, great black-backed, western, slaty-backed, lesser black-backed, and Iceland gulls. Now, Glacous Gull have been recorded breeding with some of these other species, but just typically at lower frequencies than with the American herring gull, Glaucous winged gull, and European herring gull. But so again, Glacous gulls have been cited breeding with lesser black-backed, Iceland, slaty-backed, and great black-backed gulls, again, just at lower frequencies. And to complicate things further, American herring gull have also been seen breeding with kelp gulls. These are called chandeleur gulls. And they breed with great blackbacks. These are called Great Lakes gulls. Glaucous winged gulls breed with westerns, and these are Olympic. And European herring gull breed with lesser blackbacked, and these are called Appledores. I know this is getting crazy, but I'm not even done yet. So American herring gull will also breed with slaty-backed and Iceland will breed with Glaucous winged gull, European herring gull, and lesser blackbacked. If you are starting to feel overwhelmed, then good, that is my goal. So you can see just the absolute insanity of this species complex. And this is by no means an exhaustive list of all the hybrid connections. So as I mentioned before, this species complex has 22 to 23 species of gull, and I've only named 10 so far. So yeah, I just wanted to give you an idea of just the absolute insanity of this complex. And to make matters even more complicated, it's apparent that offspring between Glacous Gull and these other birds are both viable and fertile, and they appear to have similar fitness with their parental species. So what this means is that first generation or F1 hybrids can breed with other F1 hybrids and have fully viable and fertile offspring, or F2s, or they could back cross to either of their parental species and again experience no declines in fitness. And so with that in mind, let's talk a little bit about how you might recognize a hybrid. So Glacous gulls are identifiable for their size and their coloration. So in the picture there, this is unmistakably a Glacous gull. Look at the size of it compared to that murre that it's chasing. And those pale white feathers and the pale gray on its mantle make it a very distinctive Glacous gull. And hybrids are identified in a lot of the same ways based on morphological traits that are intermediate between Glacous gull and other species. So for instance, in this picture, we see a typical Glacous gull with the pale gray feathers and then the white tips on the primaries. In this next picture, we see a typical European herring gull with black coloration on the primary feathers. And this would be a morphological hybrid between the two. You can imagine with this absolutely insane, intense web of hybrid connections that recognizing hybrids really isn't always so easy. And furthermore, each of these different species has a fair amount of morphological variation that's accepted, and those morphological variations tend to overlap as well. So you might have a bird that has intermediate traits, and it could be a hybrid, but it could also just be a bird with sort of non-average measurements. And so this extent of hybridization in the complex, as well as their recent origin, have made it really challenging to determine the phylogenetic relationships between Glacous gull and other species. But some previous work has been done. So in the 1990s, using allozymes, one researcher found that Glacous gull were more similar to species of other gull who were breeding at the same site than they were to other Glacous gull breeding at other sites, and that this might even be more prevalent at higher latitudes. Other research done by Liebers et al. And Sternkopf et al. Using mitochondrial data found that Glacous gulls might be biphyletic. So what they'd found was that European Glacous gull had one mitochondrial haplotype, and North American Glacous gull had another mitochondrial haplotype. And Sternkopf suggested that maybe after the last glacial maxima, Glacous gull, which radiated into Europe, may have interbred with the European herring gulls and undergone a full mitochondrial replacement, and that might be why there's this European mitochondrial haplotype. And basically all the research that I found too seems to suggest that Glacous gull are more closely related to American and European herring gull than they are to Glacous winged gull. So the main objective of my project is to look at population genomic differentiation in Glacous gulls. But I really had to look at this hybrid question first, because my concern was that I might bias estimates of population differentiation if I accidentally included hybrids. And since the hybrids are so visually challenging to identify, I needed to identify them using genetic means. So the first step of my project was sampling and sequencing. And with the help of several international collaborators, I was able to compile 196 samples. So that included 15 Glacous winged gull, 20 American herring gull, 24 European herring gull, and 137 Glacous gull. And I chose to sample these three species again because of the frequency of hybridization between them and the Glacous gull, and just for my own sanity and logistically, it was just impossible for me to sample all the potential gulls that Glacous gull might be interbreeding with. So the next step was DNA extraction. We used a modified salt extraction protocol with an ethanol precipitation step. And a lot of my samples were liver tissues, so they were quite fatty, and a lot of them also required a chloroform step to help break down the fat. And it's here I need to thank Alex and Arjun, who were my wonderful SWEP students, who did a lot of this work with me. Next up, we had library preparation, which was done by Jen Sin here at Queen's. We used SBF1 and MLUC1 and 200 to 400 base pair fragments. And double digest restriction site associated DNA sequencing, or DDRAD sequencing, was done at the Center for Applied Genomics in Toronto. And it's here that I need to pause and I need to thank Jensen for his incredible foresight. So in February, I'd finished my lab work, I was feeling good, I was getting ready for a field course in Brazil, and I talked to Jensen and he'd given me an estimate for my library when it would be done, which would be partway through the time I was in Brazil for the field course. And I said that it wasn't a big deal, that I could just ship my samples in early March. And Obviously, Jensen and I, at the time, had no idea what was coming in early March, but I think you guys all have an idea of what was coming. And so at the time, Jensen, he said, you know, oh, if I put in a couple of extra hours, I can get it done early and we can ship before Brazil. And I kind of said, don't stress, like there's absolutely no rush, but like if you want to, go ahead. And he was true to his word. He finished the library prep early, and I was able to ship my samples on February 10th, which was a couple days before I went to Brazil. Side note, Brazil was absolutely wonderful. We saw amazing wildlife. Thank you so much, Steve. But while we were there, we had very limited internet. And so Jensen and Vicky had to step up again, and they answered questions that the Center for Applied Genomics had before they could go through with my sequencing. And I got back from Brazil in early March, and within days, things started to shut down due to the coronavirus pandemic. And not long afterwards, I got an email from the Center for Applied Genomics saying that they were going to finish up a couple of the jobs in their queue, and then they were going to switch over to priority COVID-19 related sequencing only. And thanks to Jensen's insistence on shipping before Brazil, I had my data back on March 27th. And I can say for sure that I wouldn't be presenting results for you today if it wasn't for Jensen's incredible foresight. So thank you so much. It's been hard enough finishing a master's during a global pandemic, but I know it wouldn't have been possible if you hadn't made sure to have my samples done before Brazil. So thank you so much, Jensen. The next stage of my project was data filtering. So first, I assessed the sequence quality using FastQC, and PCR duplicates were removed using a custom Python script. Demultiplexing was done with process rad tags in stacks, and then reads were aligned to an American herring gull reference genome using BWA-MEM, and the alignment success was measured using SAM tools. Aligned reads were assembled in stacks using RefMap and populations, and finally quality filtering was done in VCF tools. And so as I mentioned before, ddRAD sequencing requires really high quality samples in order to generate sufficient data. Unfortunately, many of my samples were not high quality. Most of my samples were older than I am, and many of my samples were actually were sampled in the 1960s and early 1970s. So unfortunately, I experienced a 50% loss in my samples through data filtering. And this wasn't completely unexpected. I knew the age of some of my samples. I knew that they weren't all of the highest quality, but this 50% loss was still just absolutely devastating. So what I had in the end was 98 samples, including 15 Glacous winged gull, 18 American herring gull, 6 European herring gull, and 59 Glacous gull. And this data set with all 4 species had 2,145 loci. And this is a sampling map here of all 4 species, which you can see in the different colors. So you can see I've got 3 populations of Glacous winged gull, 2 in Alaska and 1 in British Columbia. That's in the pink. 2 populations of European herring gull, 1 from Newfoundland and 1 from Nunavut, and 1 population of European herring gull from Iceland. And then 9 Glacous gull colonies. But a couple of these, you can see I haven't named them just because I ended up with only 1 sample from each of those 3 locations. And so moving forward, there was sort of a limited amount of work I could do with them. So now we get into the looking for hybrids. So some of the programs I used to look for hybridization in my data set were principal component analysis, or PCA, principal coordinates analysis, or PCOA, structure, and new hybrids. So I used PCA and PCOA to visualize the data from all 4 species in multidimensional space. And what these methods do is they preserve the variability of the original data set whilst finding linear variables to explain aspects of the data. And they cluster samples slightly differently. So PCA finds correlations among samples, and PCOA uses distance among samples. But overall, when you're looking at either of these plots, samples that are grouping more closely together are more genetically similar. So looking at some of my data now, on the first axis, on the x-axis, we see principal component 1, which explains about 10% of the variation. And on the y-axis, we see principal component 2, explaining less than 4%. And the colors are representing my 4 species: Glacous, Glacous winged, American herring, and European herring. And what we're seeing in this plot is that the Glacous winged gulls in pink are separating far out on the first axis from the Glacous gulls and the European herring gulls. And we're seeing the American herring gulls in yellow separating out on the second axis. but we're not really seeing any separation at all between Glacous Gull and European herring gull. So that's between the blue dots and the orange. If we take a look at the next axis, PC3, we see that it's not explaining very much variation at all, less than 3%. And we don't really seem to see any further clarification on the relationships between these species. If we go to look at the principal coordinates analysis, we see a really similar pattern. So again, the glaucous winged gulls in pink are separating far out on that first axis, this time explaining more than 15% of the variation. And we see the American herring gulls in yellow separating out on the second axis, this time explaining a little more than 5% of the variation. And again, this close grouping between European herring gulls and glaucous gulls. And if we look at that third axis, it explains less than 3% of the variation. And again, no further clarification on the relationship between these four species. Now, some eagle-eyed viewers may have noticed that some hybrids are visible in these plots. So when we put the PCA and the PCOA side by side here, we see that there are 3 glaucous gull appearing amongst the American herring gulls. Those are the 3 blue dots clustered in amongst the yellow. And we've also got 1 European herring gull appearing with the American herring gulls. So that's the 1 orange dot amidst the yellow, and 1 American herring gull clustering with the glaucous winged gulls. That's the yellow dot in amongst the pink. The next method I used was structure. Structure is a Bayesian clustering program, and it assigns sampled individuals to genetic populations by minimizing deviations from Hardy-Weinberg equilibrium and linkage disequilibrium within the populations. And I used one model for this section, so I used the priors model, which allows structure to use the sampling locations while it clusters. In this case, rather than using sampling locations, I told structure the species designations. And it assumes that the sampling locations, or again, in this case, the species designations, might be informative, and it uses them while it's clustering. And the priors method is, of the models available within structure, it's the most able to detect subtle structure. I also used 4 methods to find the best K. So this is the best number of genetic clusters: Avano, Pritchard, parsimony, and visual analysis. Each of those first 3 methods have their pros and cons and their strengths and weaknesses. And so using visual analysis alongside those is really important to determine if any of them have determined the best K. So this is an example of what a structure plot might look like. This is not my data. So on the x-axis, we have each vertical bar representing an individual gull. And on the y-axis, we have the probability of membership in the genetic population. And in this case, in this example, the genetic populations have been set to 4. So K is equal to 4. And each of the colors represents one of those 4 genetic populations. So again, in this example, we see some pure glaucous gull individuals, some pure members of each of the other species, and some potential hybrids between the 2. We move to look at my actual data. So this is with the priors model and my 4 species: glaucous winged gull, American herring gull, European herring gull, and glaucous gull on the x-axis there. And K 2, 3, and 4 were the Ks that were selected to be the best by those methods that I mentioned before. If we look at the K is equal to 2 plot, we see that the glaucous winged gulls very clearly separate out from the other species. If we look at the K of 3 plot, we see that glaucous winged gulls are clearly separating, and so are the American herring gulls in light purple. But when we go to the K of 4 plot, we see that structure seems to be unable to separate the European herring gull from the glaucous gull, and is instead showing some maybe internal glaucous gull structure in the green. We are seeing some hybrids appearing in these plots though. So we have those same 3 glaucous gull individuals that appeared in the PCAs and the PCOAs that are appearing here in light purple. And we've also got these other 2 individuals, the ones we saw before: the American herring gull with the glaucous winged gulls and the European herring gull with the American herring gulls. Now, something I was a bit concerned about was that maybe the strength of the differentiation between glaucous winged gulls and the other species was overpowering the differentiation between the others. So I also ran structure without the glaucous winged gulls. So that's what we see here. On the x-axis, we've got American herring gull, European herring gull, and glaucous gull. And we're showing a K of 2 and a K of 3. And you can see in the K of 2 plot that the American herring gull are separating out very cleanly from the other 2 species. But again, in the K of 3 plot, structure is unable to differentiate between glaucous gull and European herring gull. And we've again got those 3 glaucous gull hybrids that are appearing, and those same 2, the glaucous winged gull hybrid with the American herring gull, and the European herring gull hybrid with the American herring gull. So the final method I used to look for hybrids was this program called New Hybrids. New Hybrids is unable to handle really large numbers of loci. So what I did was I took my 2,145 loci and I subsampled them 10 times, each time sampling 500 loci. And on the x-axis here in this example plot, we have vertical bars, again representing individual gulls. And on the y-axis, we have the probability of membership in one of these 6 hybrid categories. So that could be pure Glacous Gull or pure member of one of the other species, F1 or F2 hybrids, and back crosses to Glacous or the other species. And these plots are read really similarly to structure, but it's important to note the differences. So structure shows the probability of membership in a genetic population, where New Hybrids shows probability of membership in one of those 6 hybrid categories. So looking at my data here, we have Glacous and Glacous winged gulls in this plot. And we can see that the Glacous are appearing really cleanly in blue, and the Glacous winged gulls are appearing cleanly in red. And there's a couple individuals in the yellow and the black that appear to be back crosses between Glacous and Glacous winged gull. If we look at the Glacous and American herring gull plots, we start to see a problem. So you see that the Glacous gulls are all appearing in blue, but we don't have any American herring gull appearing in red. Instead, all of them are appearing in green, which are considered F2 hybrids. So New Hybrid seems unable to identify pure American herring gulls. We see the same thing again when I ran Glacous and a pooled data set of American and European herring gulls together. Again, we don't have any individuals appearing in red. We're seeing them all designated as F2 hybrids. And finally, I ran Glacous Gull and European herring gull, and New Hybrids was unable to identify really any differentiation between these 2 species, and instead designated almost all of the gulls as F2 hybrids. So what we're seeing here is a lack of convergence, or a failure to converge on the part of New Hybrids. And I tried 10,000 burn-in and 100,000 repetitions, 20,000 burn-in and 200,000 repetitions, and a reduced panel of high FST SNPs. And this is what I saw. And so what this means is that there's not a lot we can take away from New Hybrids for these analyses, since it's unable to differentiate between the species using the 500 loci. So to summarize my search for hybrids, I used PCA, PCOA, structure, and New Hybrids. And overall, these methods consistently struggled to separate Glacous Gull, especially separate Glacous Gull from European herring gull. But New Hybrids especially struggled to separate Glacous Gull from any of the other species, save the Glacous winged. But overall, through comparing these 4 methods and the results that I was seeing, I did find that 3 hybrid individuals were consistently identified by most or all of the analyses. So this is what I moved forward with. So now I was able to get to my real question, which is population genomic structure in Glacous Gull. So I removed those 3 putative hybrids, and I removed the other 3 species so that I could focus solely on the Glacous Gull. And this is a sampling map here of my Glacous Gull colonies. So the orange is the breeding range, and the blue are the sampling sites. Now, 2 of the Glacous Gull hybrids were from Baffin Island, and 1 of the Glacous Gull hybrids was from Carrick Lake. And since Baffin Island already only had 4 individuals from it, and then 2 hybrids were being taken out, I just took away that entire population because 2 wasn't quite enough to go forward with. So moving forward, my data set was 54 individuals and 621 loci. To explore population structure, I used some of the same analyses that you've seen before: principal component analysis, or PCA, principal coordinates analysis, or PCOA, and structure. But I also used pairwise FST, and I used Bayescan to look for outlier loci. So here is a PCA of our Glacous gulls. So again, we've got 5 populations: Carrick Lake, Akbatok Island, and Devil Island, all in Nunavut, Bear Island or Bjornoya in Norway, and Bjarnarhafnarfjall from Iceland. And on the first axis, the PC1, we're seeing less than 5% of the variation explained, and in the second PC, just over 4%. And the biggest takeaway that I see here is these outliers in blue appearing on the first axis, separating far from the rest of the group. And if we move to look at the second axis, we see not much population differentiation and not much more variation described by that third axis, less than 4%. Now, if you really squint your eyes and look at this plot, you'll kind of see that the orange and the green dots, which are Bear Island and Bjarne Hafnarfjall, those are our European populations, and they're clustering a little bit to the right-hand side of this plot. And the North American, which are the black, gray, and blue, are clustering a little bit to the left-hand side of the plot, and those are our North American populations. And we see a similar pattern emerging, excuse me, if we look at the principal coordinates analysis, with slightly more of the variation explained on the first axis, about 6% or 5.5%. And again, we're seeing a little bit of this European North American divide, with Bear Island and Bjarne Hafnarfjall appearing on the right-hand side, and our North American populations on the left-hand side. And again, when we look at that third axis, when we bring it in, it describes less than 4% of the variation, and we're not seeing any further separation between these populations with this extra axis. So next, I used structure, and this time I used 3 different models to look for population differentiation. So the admixture model allows structure to consider that an individual may have inherited a portion of their genome from several ancestral populations. And this more closely models sort of real-world data, but it's not as good at detecting subtle structure as the Priors model was. And the Priors model, again, as a reminder, is able to use the sampling locations when it's clustering. And it's the most powerful model at detecting subtle structure. And again, I used 4 methods to find the best K, or the best number of genetic clusters: Ovano, Pritchard, parsimony, and visual analysis. So this is an example of a structure plot, again, not my data. So on the x-axis, we've got each of these 5 theoretical populations, and on the y-axis, the probability of membership in a genetic population. In this case, we've got a K of 5. So this would be an example of very, very clear population structure, where each of the sampled populations is also a genetic population. And this is another example plot with K of 2. And what we're seeing here is that almost all of the individuals have about 50% membership to one population and 50% membership to another population. And what this suggests is that, in truth, there's no population genetic structure present in this plot. So a K of 1 would be the true reading. And so now let's look at my actual data. So these are plots run with the admixture model. And again, on the x-axis, we've got my 5 populations: Bear Island, Bjarne Hafnarfjall, Carrick Lake, Akbatok Island, and Devil Island. And a K of 2, 3, and 7 were those selected as the best by those models that or the methods I mentioned earlier. And if we look at the K of 2 and the K of 3 plot, we're seeing that this isn't very indicative of population genetic structure. We're seeing that that all of the individuals don't have full membership to any of the genetic populations. And this suggests that a K of 1 is the true reading. But when we look at the K of 7 plot, we do see that there seems to be a bit of a divide between the European and the North American populations, with the European populations in the light purple and the North American populations in orange. When we look at the admixture and Priors models combined, we have a similar result. K of 2, 4, and 7 were those selected as the best, but K of 2 and 4 are again showing that there really isn't a lot of population genetic structure present here. But looking at K of 7, we see this European North American divide coming through again, with the European populations mostly in yellow and the North American populations mostly in orange. Finally, when we look at the Priors model, we're seeing a really clear North American-European divide. And a reminder that this Priors model is the best at detecting the most subtle structure. And this is what's coming through most clearly. That's clear distinction between Europe and North America, suggesting weak population structure between them. Finally, I used pairwise FST. FST compares the variation in genotype frequencies within and between populations to index population genetic structure. And I found significant FST values between Bear Island or Bjornoya, Norway, and Devil Island and Carrick Lake. These values were low, but they were significant. And it may seem a bit odd that Bear Island and Devil Island are separating or are being considered differentiated when they don't seem that far apart compared to some of the other populations. But it's important to note that birds rarely ever fly straight from one population to another. And so, especially seabirds, they tend to not fly across large expanses of land. And so the true sort of flying distance between these populations might look a little bit more like this, suggesting that glaucous gulls probably don't fly across the northern part of Greenland. And finally, I did an outlier analysis to look for outliers, and I used base scan. and I didn't find any putatively adaptive loci. So what this suggests is that all 621 of my Glacous Gull loci are neutral. So to summarize what I found, I had weak population structure between North America and Europe using PCA, PCOA, structure, and pairwise FST, and I didn't find any outliers in Bayescan. And as I mentioned before, conservation units can be divided for management, right, using different combinations of loci. And those were the evolutionarily significant units, management units, and adaptive units, or ESUs, MUs, and AUs. And since I wasn't able to find any adaptive loci when I did my outlier analyses, I can't really inform evolutionarily significant units or adaptive units. But using the data that I do have, which appear to be neutral loci, I could suggest that North America and Europe be managed separately as separate management units. But there's still a lot of work left to be done. So I've had lots of wonderful people suggest further analyses to look for hybrids. For instance, I could use programs like Construct, GG Hybrids, HSAR, SplitsTree, PCAdapt, and linear discriminant analysis. I'd also like to do a little bit more analysis on population genetic structure. Specifically, I'd like to try to estimate the migration rate between my populations using Bayes-Ass. And I'd like to look for correlations between geographic and genetic distance using a Mantell test. And this could suggest that rather than sort of true population genetic structure, what I'm seeing might be more of a pattern of isolation by distance. So to conclude, my work is the first that uses high throughput sequencing technologies on population genetic structure in Glacous Gull. And mine's the first work to find this North American-European divide in nuclear loci. As I mentioned earlier, it has been found in mitochondrial DNA, but not previously in genetic DNA. And my work also mirrors some of the previous work that was done, which finds that Glacous Gull are more similar to American and European herring gull than they are to the Glacous winged gulls. Another big takeaway from my project is the need for a greater contemporary sampling effort. So I sent 196 samples for sequencing, and less than 40% of them were sampled in this century, so in the last 20 years. So my youngest sample was 7 years old, and my oldest sample was 55 years old. It's incredibly challenging to get high quality dd-rad sequencing or whole genome sequencing quality data sets out of samples that were taken before the invention of PCR. If we're going to try to conserve genetic diversity, which we absolutely should, then we desperately need contemporary estimates of population genomic structure, and for that we need samples. And finally, Glacous Gull are important members of Arctic ecosystems. As an apex predator, they perform all kinds of important ecosystem services, and they're a valuable bioindicator species. There's been more than 60 papers published on Glacous gulls as a bioindicator in the Svalbard archipelago alone. And they are a traditional food source in Alaska, Canada, Iceland, and across the circumpolar Arctic. And they deserve an informed and a proactive management strategy to keep this gull conserved for many, many years to come. And with that, I'd like to thank Friesen Lab members, past and present, 50 years worth of sample collectors, my SWEP students, Arjun and Alex, committee members, and all the organizations that provided funding and samples for this project. Thank you all so much.
Thanks so much, Emma. That was a really nice presentation. You could pronounce it with the Icelandic line. That was awesome. So at this point, then, I guess I better preface by saying my internet connection is unstable, so if I fade in and out, let me know. I'll read any questions for now, but if I do fade away, then Emma, maybe you better take over.
Okay, I'm ready.
Okay. So, questions for Emma? People are busy typing. Okay, from Bill Nelson. Thank you for an interesting talk. If I recall correctly, you were saying that genotyping was done using gull livers. Now that you've done this work, can you use it to develop a less intensive genotyping approach that can identify hybrids in this species using non-destructive samples? I'm curious because it would be interesting to know what the genetic structure looks like across more populations and, of course, in more recipient species samples, more recent samples. Emma.
Yeah, so it was sort of just sort of coincidence that a lot of my samples were liver. It just depended on where I got the samples from, but like non-invasive blood sampling would have been just as effective, and again, probably more effective if it was done more recently and, you know, preserved at good temperatures like -70 right away. So it really doesn't have to be a sample that actually kills the bird, which a lot of my samples unfortunately were. But yeah, it's completely possible, and it happens all the time that you can have non-invasive blood sampling methods that are able to generate really, really high quality DNA. So yeah, unfortunately, a lot of my samples were killed in the process, but it doesn't have to be that way.
Thanks. So from Chris Eckert, do you think that mapping your reads to the herring gull genome created a bias in the SNPs that you analyzed? Were there differences in the percent of reads mapping to the reference among species?
That's a good question. I'm not sure about if some mapped better than others, but I have wondered about that a little bit. Because again, it was the American herring gull that I used as the reference genome, so that yeah, maybe that biased and made my herring gulls harder to tell apart from the Glacous gull for that reason. But I guess I don't really know quite what I could have done differently. Like, I don't have access to a Glacous gull reference genome, and that really would have been ideal. So the American herring gull was the closest I could get. So yeah, I'll have to look and see if maybe some of the species map more closely than others. But yeah, you're probably right that especially something like the Glaucous winged gull would group like less well to the American herring gull reference genome than some of the other species did.
Okay, thank you. Next question is from Steve Lougheed. He says, Emma, 3 hybrids that you identified using all methods showed no admixture whatsoever, I think, and a new hybrid were considered back crosses. What is the possibility that these were simply misidentified? Did you have mitochondrial DNA as well?
Yeah, so that's a good point. So in the hybridization plots I showed, those were all run with the Priors model, which I think is like, it's typical that it tends to show sort of complete or like almost 100% membership into one population or the other. So I think that's partly why they appeared so strongly as, you know, American herring gulls in that case. But I think you make a fair point there in that it's possible that some of these gulls were just not recognized as or incorrectly identified as Glacous gull when in reality they were something else. But I do think, like, for people who are doing sampling up in the Arctic, I personally haven't seen Glacous gull in person, but I've heard that they are quite distinctive, and the size difference between them and a typical American herring gull would be quite severe. And again, American herring gulls tend to have black on the primaries. So yeah, it's possible that some of these gulls were just unusual and maybe didn't represent true hybrids, but I think based off of other studies that show how frequent hybridization is, it's probable that some of these were hybrids in reality.
Okay. Oh, now there lots of questions coming in. Okay, the next one is from Sarah Gordon. This was such an exciting presentation. I just wondered if you could expand on the contaminant risk in the gull as an apex predator. You mentioned accumulation as they're at the top of the food web, which I thought was biomagnification. But was there also evidence of bioaccumulation in the population as well, i.e. historic increase in contaminants in this species in general?
Oh, that's a good question. I don't know the answer to that right away. I'd have to double check the studies that I've read, I may have just misspoken by using bioaccumulation rather than biomagnification. So yeah, I'd have to double check some of those studies. But yeah, I have seen just that the Glacous Gull tend to have much higher contaminant loads than other species, which I guess is fairly typical for apex predators.
Okay, Chris Eckert. Oh, I'll just follow up. Sarah Gordon followed up by saying, I get confused with those terms all the time too. Okay, Chris Eckert, if I gave you a random gull liver, how confidently could you assign it to a species or population based on your data? You could answer this with k-fold cross-validation.
Yeah, I guess not super confidently. I think, yeah, if it was a glaucous winged gull, we could pretty confidently identify it or separate it from the glaucous gulls. But definitely with the European herring gull samples that I had, we really wouldn't be able to tell them apart from the glaucous gulls. And maybe it's partly just that I only had 6 European herring gull samples. And again, one of them was identified as a potential hybrid. So maybe if I had more samples from areas where glaucous gulls don't interbreed with European herring gulls, maybe I would have had better success. But yeah, I think you make a strong point there that I probably wouldn't be able to separate glaucous gull from European herring gull for sure, and maybe from some American herring gulls if you just handed me a blood sample and I had to run with it.
So Emma, follow-up question from me. Are you comfortable that glaucous gulls are a good species? And by what definition would you call them a good species, if any?
Yeah, so that's something I've consistently struggled with for the entire last 2 years. Obviously, some definitions of species mean that they shouldn't be hybridizing at all with other species, that would make them not a species. So yeah, I don't entirely know. There's definitely morphological variation between all of the 22 to 23 species in this complex, and from spending time on North American Gulls, the Facebook group, everyone there seems pretty confident when they're identifying gulls or identifying hybrids, but it's all based on morphological variation. And as I said, there's so much overlap. So yeah, I guess, yeah, I don't really know. It's something I just consistently struggle with.
Yeah, not just you. It's been a mess for a long, long time.
Yeah.
Jen Provencher followed up. Biomagnification is across food webs and bioaccumulation is with age. Thank you, Jen.
Okay.
I didn't know that, so that's good to know. So I'm not seeing, there's a couple more minutes if anyone's got another question. Greg Robertson, thanks for joining us, Greg. Excellent presentation, Emma, both delivery and content. No putative adaptive loci at all. Is that unusual?
Yeah, so I was a bit surprised by that. But I think, at least from talking to Lily, who was also in our lab, she did her project on Northern Fulmar, and she also didn't find any adaptive loci. And she had more loci in total than I had. So yeah, maybe it's not super strange. I don't really know. I was disappointed for sure. I was really hoping to be able to find some adaptive loci and maybe get into looking at adaptive units. But yeah, I'm not quite sure how unusual it is to find no adaptive loci with an outlier analysis like Bayescan.
Okay, and just Jen has a comment which is probably good to end on. Also important to note that Environment and Climate Change Canada and partners had these samples for contaminants work. No birds were actively killed for the study. The study took advantage of these samples. Great talk, Emma. And Chris Eckert says, thanks for the interesting talk, Emma. And Steve Lougheed says, thanks, Emma, great job. And yeah, I'll echo that. That was a lovely talk, Emma. Thanks very much.
Thank you, everyone.
All right, I think you're off the hook, Emma.
Okay.